Hi everybody, today we are going to focus on the muscular system. This is the second part of our muscular system lecture. So to review what we discussed during our class last time, one of the topics was on connective tissue that is present within the muscles. And we said there are three different types of connective tissue. We have the outer most layer of connective tissue that is wrapped around your um, whole muscle, and that is referred to as the epimysium or epimysium. We have within the whole muscle, we have smaller structures called fascicles, and these fascicles are wrapped with a connective tissue called perimysium. And within these fascicles, within these units, I have groups of individual muscle fibers, which are basically my muscle cells, that are wrapped by themselves on their own with a connective tissue called endomysium. So looking at the picture here, again, kind of highlighting the things that we already seen. Right here is my muscle fiber as a whole. What I pulled out basically is a muscle fiber. The layer that you see around this kind of clear lining, which is forming these boundaries right here, would be your endomysium. If I take individual muscle fibers, right, and bundle them all together to form this larger structure, the larger structure is called the fascicle. And again, the connective tissue that you see wrapped around the fascicle is the perimysium. And I can take many fascicles, which are basically what you see here as these outlines right here. If I put a bunch of fascicles or group of fascicles together, what I create is this largest structure we have, which is my whole muscle. Let's say, for instance, your biceps, and that is wrapped with a third connective tissue called the epimysium. Please do notice that epimysium continues and forms a point of attachment to your muscle, to your bone, and that's basically called the tendon. So epimysium is the connective tissue that becomes your tendon. Here's another depiction again. Individually pull out, that's your muscle fiber wrapped with endomysium. Group of muscle fibers together form a fascicle that is wrapped with perimysium. And a group of fascicles forms your whole muscle, which is wrapped with epimysium, which connects also the bone and muscle together through a structure forming tendon. Now what I'm going to do is I'm going to show you a picture of what a muscle cell looks like. So we of course have slides on this that have basically the information nicely presented, but it's always good to kind of get an idea or the simplified version first. So if you imagine again the shape of your muscle fiber, and sorry, my drawing might not be the best drawing, but individual muscle fiber. So what I'm depicting here is a muscle fiber. Within this muscle fiber, we have the membrane of the muscle fiber, which is called the sarcolemma. And the space inside of it is called sarcoplasm. If you guys recall, the word sarco implies muscle. So sarcolemma basically is the plasma membrane, and sarcoplasm is the space within that. Now, if you look inside the muscle fiber, you will have these elongated structures that are basically form our contractile unit eventually, called the myofibril. So I'm just going to draw, well, as closely as I can, a few of these, but you will have hundreds of these myofibrils inside a muscle fiber. So again, I'm color coding you guys so you can follow these. So these are myofibrils. I can spell. And myofibrils are broken down to these vertical units, which I have right here. And these vertical units are identified as sarcomere. So each of these units you see right here within the myofibrils are called sarcomere, which are defined as my contractile units. So if you're looking at basically your muscle fiber, which is basically a cell, within that you have myofibril, and myofibril is divided to individual structures called sarcomeres, and sarcomeres are basically going to be your contractile unit. Now, what do we have in terms of sarcomere? Is what do we have inside the sarcomere? Is we have what we identified as thick filament, and I'm just gonna again try to color code it so you guys can follow it. And again, this is gonna be repeated in each unit of sarcomere, but we're only gonna be able to draw a few of them because otherwise it takes too long. So what I have depicted in green, these are called myosin, which because they are thicker, we call them thick filament. And I'll tell you why they are thick, or in relation to what they are considered thick. So the second type of protein that we have in here are your actin. So notice again the thickness of the actin and myosin. Actin is, which is the one that I'm drawing right now, in bluish color or cyan. These are thinner than what we have for the myosin. So the blue ones that I'm drawing are called actin. And again, these are going to be thin filaments. So kind of reviewing what we have learned so far. And again, it's a good way of kind of imagining going from the smallest structure to the largest structure. At the most basic level, we have two proteins which are depicted here. These are your myosin and actin. And again, I want you guys to remember that these are proteins, okay? And they're also referred to as myofilaments, okay? Now, actin and myosin are packed into individual units called sarcomere, which we're gonna take a look at it in more detail in a few minutes. Sarcomeres are, would form your myofibrils. Myofibrils are packed inside my muscle fiber, right? Which are basically cells, right? These cells are found inside the structures we call fascicles, right? We're going through microscopic anatomy, and then we finally have the whole muscle. So reminding again, what are the three connective tissue we have present within this muscle tissue or muscle as an organ? Muscle fiber cells are wrapped with endomyosin, or mysium, sorry. Fascicles are wrapped with perimysium, and whole muscle is wrapped with epimysium. So again, you have it as from the smallest structure to the largest structure. And recall that epimysium also becomes tendon, which is basically how your muscle connects to the bony tissue or to your bone. Now, the next thing I want to talk to you guys about is the actual structure of a sarcomere. So let's go ahead and draw individual sarcomeres. So let's say I have these lines that are separating my sarcomere. And within that, then I will have, within my sarcomeres, I will have myosin, which are my thick filaments, right? I'm going to just draw them as these boxes, which is easier to do rather than coloring it. So these are my myosin. And then partially overlapping with this, we have our actin. So notice again, here is my actin. Notice the difference in the thickness of this component, and we'll go over this. Why is that important in terms of visualizing a skeletal muscle fiber under the microscope in a few minutes. So these green lines, sorry, these cyan line color are my actin. So let's do some labeling on this structure right now. So looking at this here, from here to here is a unit called sarcomere, right? These are my contractile units of a muscle. The boundaries for my sarcomere, which is here and here, is marked with Z disc. And guys, please remember that some of the details of this sarcomere is missing. So don't, it's not a perfect depiction of what a sarcomere looks like, but this is what I expect you guys to know at the minimal in terms of sarcomere structure. Now, if you look at this picture, you have regions within these sarcomeres that are only made up of actin. So there is no overlapping with myosin. So for instance, from here to here, from here all the way to here, and again from here to here, all you have is only and only actin fiber. So these regions are referred to as the I band. A lot of my students ask me, well, this is still actin, correct, but it's not the whole actin because it's only the portions of the actin where it's not overlapping with myosin. Now, on the other hand, what I have is the regions that are depicting are myosin, which is also overlapping with the actin. And these regions are called the A band. So right here is A band. From here to here is also the A band. Again, these regions are both what? Your myosin and the overlapping actin that they have right here. The third structure that I have is the area where I only have what? Only have myosin, no overlapping actin. So this area sits within the A band. And this is referred to as the H zone. H zone. So take a look at this structure. And again, let's try to imagine what happens when a muscle contracts. So when we talk about muscle contraction, the first thing that comes to our mind in terms of visualizing it is that the muscle gets shorter, correct? So how is this muscle gets shorter at the smallest units, which is basically at your protein or your chemical level, your molecular level. So what you have here as myosin and actin, notice how they have some overlap here. So when the muscle is contracting, the overlap you see right here becomes more. So basically actin gets pulled into the myosin. These actins you have here interact with the myosin and gets pulled inward. So as they get pulled inward, your H zone basically shrinks because now it's overlapping with the actin, so it's not pure myosin anymore. So actin gets pulled into the myosin, the overlapping region becomes more. So instead of it being just these sections here, this whole section will be overlapped with actin either from this side or from that side. So at the same time, my I band, which is only actin, is also going to shrink or shorten because the only reason that was I band, the only region that was I band was actin, pure actin, and now that actin gets pushed into or pulled into the myosin, making your I band also smaller. Now, A band was the length of the myosin, right, which also overlapped with the actin. A band during muscle contraction doesn't change because the myosin length doesn't change, right? All you're doing is you're increasing the overlap between these two proteins, but you're not really changing the length of the myosin. So if you're looking in terms of muscle contraction, an effect of this on your muscle at the most basic level, sorry guys, at the most basic level, what we have here is your I band, which is actin, is gonna get shortened because again, it gets pushed in or pulled into your myosin. The H zone, which was also only pure myosin now have overlapping actin, so that area also shortens. A band, which is basically depiction of the height of your, or the length of your myosin, stays the same. But in an overall picture, your sarcomere, which is your muscle contractile unit, is shortened. So keep this in mind. Sarcomere is shortening. So look, if sarcomere is shortening, which are making your myofibril shortens, which causes the muscle fiber to shorten, which causes the fascicle to shorten, which causes the whole muscle to shorten. So basically, by creating an overlap at the most basic level of these muscle, at the level of myofilaments, which are my actin and myosin, increasing the overlap between these two proteins cause the whole muscle in the microscopic level to shorten, which is pretty fantastic. It kind of reinforces the same idea that the changes at the molecular level can have an effect on the macroscopic or the organ level. Now, something else that I want to talk to you guys about is the whole concept of striation. This is something we also talked about previously. So striation was basically the light and dark pattern you observe on the muscle, light and dark pattern, right? So how does this light and dark pattern create it? Imagine you're putting a sample of the skeletal muscle under the microscope. Again, a skeletal muscle would be made up of individual muscle fibers, which are made up of myofibrils, which are made up of sarcomeres. And again, in terms, sarcomeres are made up of your actin and myosin. So when you look at it, and let's say, imagine the light is hitting this sample from behind your computer. Now, the regions where you have actin, these are pretty thin filaments that allows a lot of light to pass through the sample, or this part of the sample. So right here, and right here, lots of light can pass through the sample because they have the thin filaments. On the other hand, where we have myosin, myosin is a thick filament which is going to absorb a significant amount of light, and as it absorbs, basically it allows less light to pass through your specimen. So basically these regions will appear as darker sections. So what would we have if the light is passing through the actin and myosin? I will have light band, dark band, light band, dark band, light band, and again, repeat this over and over again. And that gives us what? Our striation pattern. So if I ask you guys, what is the dark band correspond to? Dark band corresponds to the A band region, and the light band corresponds to the I band, which is basically your actin section. Let's continue and look at now what is a muscle actually, what allows the muscle to actually contract. So before I go into detail about that, and again, we're kind of touching the surface right now. We're not going to go into detail about that in terms of the physiological aspect of it. We're just focusing on the anatomy. One of the first topics I always want to mention to my students is understand that a muscle contraction requires the input of your nervous system. So if you want to contract a muscle, and I'm going to go back and just write it down for you guys. So a skeletal muscle contraction requires nervous system input. So nervous system input is through structures called neurons. This should sound familiar to you guys since we already looked at nerve tissue before, and neurons being the most important type of cells within that tissue. So how does this process happen? So your neurons start to create electrical impulse, which causes electrical impulse in my muscle. And the third step that leads into muscle contraction. So neurons create the original electrical impulse. That electrical impulse transferred or creates a new electrical impulse inside the muscle fiber, and that electrical impulse leads to muscle contraction. So how does that take place? Let's look at the anatomy of the skeletal muscle fiber in a little bit more detail and in relationship with a neuron. Sorry, put this way. Okay, so we learned some of these vocabularies already. We know sarcolemma is the muscle fiber plasma membrane, which we labeled earlier. We also talked about sarcoplasm being the cytoplasm of the muscle fiber, basically the space inside the muscle fiber. This is new, but also something we have learned before. When we looked at the cell structures, one of the first things we talked about was endoplasmic reticulum, or ER, which we divided to rough ER and smooth ER. So in case of muscle, we have a specialized ER, which is called sarcoplasmic reticulum. Again, kind of highlight, instead of saying endo, we're just replacing it with sarco, which implies we're talking about muscle. Sarcoplasmic reticulum is extremely important because, first of all, it is packed with what we identify as calcium, which is Ca2+. And this calcium is extremely important for muscle contraction. Without calcium, muscle cannot contract. Now, ER is basically a network of interconnecting tubes in this case. And again, these are specialized ER network, and they are surrounding your what? Your myofibrils. Remember, myofibrils were what we depict in blue here. So imagine there is a network of connecting tubes that are wrapped themselves around the myofibrils with the goal of carrying calcium necessary for muscle contraction. The next structure we have inside the muscle fiber is, and is specialized to muscle fibers, is T-tubules. T-tubules is where your sarcolemma, which is your plasma membrane, dips down inside the cell. And the importance of this, it allows for the process of fast depolarization. So now you may not be like familiar with the word depolarization. Basically, if you want to think about it, fast depolarization means fast spread of, easiest way to say it, electrical impulses. So if I want to spread my electrical impulse, which is basically depolarizing your muscle fibers, these T-tubules are extremely important. Now, some T-tubules have these cross channels called terminal cisternae, and they happen on in pairs on either side of your T-tubules. And I'll show you guys this in just a second. So let's take a look at this. Again, try to identify the structures as we go. Try to practice to see if you guys recall the information as we discussed earlier. So right here, we have a muscle fiber. The layer that's being peeled off here, that's your sarcolemma, which is basically the plasma membrane of your muscle fiber. These are structures that I'm peeling off. These are my myofibrils, which were the long blue cylindrical structures I've drawn inside. Notice the color coding, guys. Do you see the red lines? Those are going to be your A band, which is depicting your myosin. And the blue lines we have right here, notice the thinness. Those would be your actin, which also form your I band. Now, if you look at this again, wrapped around these myofibrils, you have these blue network of structures. These are your sarcoplasmic reticulum, so sarcoplasmic reticulum. And running within or between your sarcoplasmic reticulum are these, again, invagination inside the sarcolemma, which are called the T-tubules. So what you see in light blue color, which is basically continuation of the plasma membrane dipping down. See how it continues and it starts from here. These are T-tubules for the purpose of spreading the electrical impulse rapidly through the muscle fiber. And then on either side of your T-tubules, you have the thickened portion of the sarcoplasmic reticulum, which is called the terminal cisternae. So these regions are the terminal cisternae, and this is the actual sarcoplasmic reticulum. So let's take a look at, again, more details of what we discussed already. This is review of what we learned on a drawing, and now we're going to look at it in terms of context. So myofibrils are densely packed rods-like elements that are found inside the muscle fiber. Basically what we've shown here, these are your myofibrils. A typical muscle fiber can have thousands of these myofibrils packed inside of it. So again, this depends on the size of the muscle. If the muscles are much smaller, then they could have hundreds, but if the structures are larger, they will have thousands of them. It accounts for 80% of the muscle cell volume. So literally these myofibrils take over the space inside your cell or inside your or in your cytoplasm. Now, myofibrils, we already looked at these characteristics. Again, kind of a recollection of what we talked about earlier. Sarcomeres are those vertical division of the myofibrils, which are also identified as contract units of muscle. Within the sarcomeres, we have two myofilaments, which are my actin and myosin, known as thin filament and thick filament, respectively. The pattern created by the actin and myosin space within the sarcomere, we form your striations. Now, here's a new piece of information. So we keep talking about the fact that myosin and actin interaction causes muscle contraction, correct? We said earlier that if you go back to the picture, and I want to kind of walk you guys back through so we can see the connection. I mentioned here that actin gets pulled inside the myosin, right? Actin gets pulled inside the myosin, which basically causes the whole sarcomere to shorten, right? Now, there's a couple of questions here. First of all, how does the actin and myosin are interacting, which allows the actin to be pulled inside the myosin? So right now, you see there is no interaction based on the picture that I've drawn here. And the second question is, well, how is it that not every single muscle on my body continuously contracting if they are going to be in connection with one another? How does it act? If actin and myosin, basically, let me say it one more time. If actin and myosin are my reason for having muscle contraction, what prevents every muscle of my body to be contracting every single second of every day? What prevents it? What prevents these myosin and actin interacting until I get the proper signal from my neuron, which causes the electrical impulse in my muscle cell, which causes muscle contraction? So what is happening to a muscle when it's relaxed? How are the actin and myosin are in relation to each other when the muscle is relaxed? And how is that relationship between actin and myosin changes when there is the electrical impulse that is allowing the muscle to contract? So let's go ahead and take a look at that. So here is a depiction of actin and myosin. First of all, what you see here, this is your myosin. As you can see, the color coding. Coming off of these myosin, you have quite a few structures extending, which are identified as myosin heads. Now, myosin heads are going to interact with what we identify in blue as your actin. Now, notice there is an interaction here, right? So how does those two interact? So if you look at the actin, which is depicted as a zoom-in version here, you have these sites right here inside the actin, this little indentation. And these little indentations are referred to, these little indentation inside are referred to as myosin binding site. So basically these little indentations are what allows the actin and myosin heads to interact with each other. And notice that these myosin heads, they have these little arrows, right? These arrows means that the myosin head is going to pull the actin inside itself. So it kind of, you know, like the tug of war. Imagine this is your rope and these little myosin heads are your hands. You grab the rope and you pull it in, right? Then you release it, which is myosin head separating from your actin. You grab another piece of rope and then again you pull it, right? So these motions, kind of think about what your hands will be doing. The myosin head will be creating the same motion. So that answers the first question. How is the actin and myosin are actually interacting that causes the muscle contraction? They do that by myosin heads interacting with the myosin binding site. And through this interaction, you're allowing the actin to be pulled into the myosin. The second question, however, was, how is it that not every single muscle in my body is contracting every single second of the day? For that, we are going to talk about two main proteins, which are the ones that basically make sure muscle is not contracting in the absence of the electrical impulse. These two proteins called troponin and tropomyosin. These are regulatory proteins that ensure that the actin and myosin cannot interact. But how are they doing that exactly? Let's take a look at this picture, guys. See these little bumps we said, these little indentations that you see in black right here? These are my myosin binding site. Now notice how there is that kind of a strip of orange that goes right over and cover all of the binding sites. Those are called what? Tropomyosin. Now you also have these little circles. These are individual subunits which together form another protein called the troponin. So right now in this picture, troponin and tropomyosin are working together to cover what? To cover the myosin binding site on your actin, which prevents the actin and myosin to interact with each other. We already talked about this, I'm just gonna skip these. So here again, depiction of the same concept, myosin and actin interacting with each other. Every time the muscle contracts again, these little heads are gonna shift, right? Actin gets pulled inside, but as it gets pulled inside, the whole sarcomere is shortens, which basically implies that your whole muscle will shorten in the big picture. So let's go ahead and now connect the concept of the nervous system to our skeletal muscle. Skeletal muscle fibers and muscle contraction. So we already talked about it. I already wrote it down for you, but reviewing it again. A skeletal muscle contraction requires the input of the nervous system. Nervous systems are made up of cells called neurons or nerve cells. And nervous system are communicating with the skeletal muscle through what again? Electrical impulses that originate from your neuron. So we're gonna talk about the neuron or nerve cell structure. And I'm gonna end the lecture and we'll pick up the rest of this in our next PowerPoint. So a nervous structure or nerve cell structure is the following. You have these many extensions or processes right here called the dendrites. These are identified as the receptive side of your neuron, which is where basically they receive electrical stimulation from the previous neuron and then pass that electrical impulse through the next structure, which is your cell body. So these arrows are basically depiction of the electrical impulses. They come from the dendrites and they pass it to this central section, which is your cell body. What's the importance of cell body? Cell body produce a specific structures called neurons. Produce a specific proteins called neurotransmitters. And neurotransmitters are basically allow for communication. Sorry, I can't spell clearly. Communication. So the last part of a neuron is this long extension you see right here, which is called the axon. So axon takes the electrical impulse from the cell body and pass it again through its length until it gets to the end of it, which is your axon terminal. What is the importance of axon terminal? Axon terminal is important because it is where you store the neurotransmitter that were produced by the cell body. So looking at this as an overview, a neuron is essential for electrical impulses or creation of the electrical impulses that leads to muscle contraction. These electrical impulses start with your dendrites, which gets passed down to the cell body, to the axon and axon terminal. Axon terminals is important in terms of storing the neurotransmitters that were originally produced by the cell body. And these neurotransmitters are basically going to be the communicators or the molecules that allow a neuron to communicate with your muscle fiber, which eventually again leads to muscle contraction. Okay, so let's stop here and then we'll continue this PowerPoint on our next presentation.